So good morning, everybody. It's nice to launch this session for the press on the things that are happening in the world of clinical translation for stem cell technologies. My name is Chuck Murry. I'm the director of the Institute for Stem Cell and Regenerative Medicine at the University of Washington. To my right, the panelists are Sally Temple from the Neural Stem Cell Institute, David Lebwohl from Semma Therapeutics, and Vivian Tabora, who's not yet connected with the company, I guess, perhaps too much, but is leading the Parkinson program at the Memorial Sloan-Kettering Cancer Institute that's being done in collaboration with Cornell University. And so I'd like to just make a few opening remarks, and then we'll turn to each of the panel members and let them do some presentations. But I'd like to start by quoting one of my favorite Nobel laureates, and that is Bob Dylan, who tells us that the times they are a-changing. And it's been a very interesting trajectory for cellular therapy, particularly for work that's being done with human pluripotent stem cells. It's shown promise for 20 years, right? It's been over 20 years now that we have had these technologies in our hand. And the trick has been that the human mind can race ahead to the potential of these cells to do all kinds of societal good, but the reduction to practice has been slow. And so I would submit that this is a revolution in medicine that's happening, but sometimes slow revolutions are hard to really appreciate. I mean, if we look at the way therapeutics have evolved, we had a very quick one many years ago with small molecules, development of antibiotics, small molecule antagonists, things like that. These were really things that chemists could make in a test tube. The next wave really began perhaps in the late 1970s as molecular biology came to the fore, and we began to use large molecules as therapeutics, antibodies and things like that. And these were molecules that were too big really to synthesize in a test tube, and so consequently we used cells to manufacture them. The third wave, which I think will become the third pillar of medicine, is using cells themselves as therapeutics. And this is so much more complicated, of course, to reduce to practice than just the building of molecules, because while the chances of cells to do good are vast, the chances of them to cause mischief of various sorts is also fairly considerable. And so the way to use these in a controlled and responsible way, it has been quite tricky. And so that is what the people have been spending the last couple of decades doing. I'd submit, though, that a word that you're going to hear a lot at this conference is inflection. There's a growing sense of excitement that we're nearing an inflection point where it's responsible, scientifically and ethically responsible, to be taking some shots on goals in using cells as therapeutics. And we've had an area, what I call the big five areas that have emerged, and four of these will be represented today. We've got Parkinson's for dopaminergic neuron replacement. We've got diabetes type 1 with insulin-producing beta cell replacement. We've got the retina with retinal pigment epithelium. The one that's not represented today is spinal cord injury, particularly with oligodendrocyte therapy. But all of these things are either in or very close to clinical applications. The fifth of those is my area of expertise, which is in heart regeneration using cardiomyocytes. I can tell you just a few quick things that are going on in the cardiac world. Most heart failure is caused by cellular deficiency, and it's a deficiency of the muscle cells, particularly, that are the culprits behind the failing heart. And the notion was very straightforward, that we should be able to therefore use cells as therapeutics. And over the last 20 or so years, this was something that I grossly underestimated. I thought it would be done in about 5 and we'd be into the clinic, but it's been about 20 years. We've learned how to take human stem cells, grow them up in vast numbers, differentiate them into beating heart muscle in a dish or in a bioreactor. We've learned the rules of how to transplant them, keep them alive in laboratory animals, and we've sort of run our way up the food chain from rodents to guinea pigs, now to non-human primates and pigs. And a lot of lessons are emerging. One of them is that there's an extraordinary ability to do good. In our own work, we've restored the beating function of the heart essentially back to normal levels after an animal's been given a very large heart attack or myocardial infarction. And no drug therapy has ever been able to come anything close to this. And so we are extremely excited about this. Conversely, we've also uncovered an unexpected toxicity along the way, which is that these cells destabilize the heart's rhythm, and they create electrical malfunction of the heart. And so one of the toxicities, if you will, that we're working with at the moment is to reduce the amount of arrhythmias that the cells cause as they are healing in. So lessons like this tell us what do we learn from this? First, that there are going to be therapy-specific areas of toxicity that come up, but then there's also going to be more broadly based things that all cells as therapeutics are going to have to face. And these, the best example perhaps of that is what to do about the immune system. I think most people at the table would agree that at least for initial approaches, what we need to have is an off-the-shelf, what's called an allogeneic product, which is not making cells from each person to use as that person's specific therapy, but rather to have a batch of cells that we can grow up in large quantities, control their quality, make sure that they're free of genetic instability and other kinds of things that we don't want to have, and push this out into the community as a therapeutic. But that means, of course, we're going to have to deal with the immune system. Some people dwell in immune privileged compartments, perhaps, and don't have to worry about it as much. But others of us dwell in the real world, where we have to deal with immune suppression and immune mismatches. So that's a remaining challenge. But this is being tackled in several ways, as we've heard about at the meeting. So with that as sort of a preamble, I'd like to introduce my colleagues then and ask them to come up to the podium and talk about the work that they're doing. First will be Sally Temple from the Neural Stem Cell Institute in New York. And I would just remind the folks, if they could keep their remarks to 5 to 7 minutes so that we'll have some time for questions and answers at the end, that would be great. So Sally, please.
Great. Okay. Thank you. Thanks so much. And I don't know if this is on.
Yeah. It is on.
Oh, it is? So, okay, great. So thanks so much. So I'm going to talk today about where we are with retinal repair, the promises and the new therapies that we see emerging, and also some of the challenges that we're facing in this field. The unmet need is just so obvious, right? In terms of children, 700,000 children who have visual impairment. From adults, over 7 million adults with visual impairment, and I think that the effect of this on people's lives is seen when you look at the median annual earnings, around $38,000, and over 1 million people who have visual impairment who are living below the poverty line. It speaks for itself that of the need and the impact on people's lives. So one of the challenges is to create the very diverse cells that are present in the retina. So the retina, so you look through someone's pupil and you can see a black layer inside. That is actually the retinal pigment epithelium that's shown here, which is a monolayer of cells at the back of the eye that's very important for sustaining what's called the neural retina. And the neural retina includes the photoreceptors that lay right next to the retinal pigment epithelium, and also several other nervous system cell types that culminate in the retinal ganglion layer. And those retinal ganglion cells actually converge their processes and send an axon in the optic nerve that goes back to the brain. So we have a significant challenge in being able to make all of those cell types and also to reconnect with the brain. Where you have mutations, for example, in diseases like retinitis pigmentosa, there's an additional challenge of having to correct that. So the remarkable progress that has been made since the discovery of human pluripotent stem cells is that retinal progenitor cells can actually reform all of the retinal cell types, and they do it on order. It's very similar to the gestation that normally occurs. And you can see that they can create even three-dimensional masses that have layering of the key cells that are present in the retina, including the photoreceptors that form on the outside. So you can see the outer layer, and then the inner layer also contains these different neural cell types. So we can make those. There are even ways to scaffold the output and growth of these so that you can make, as you see on the right in K, these almost fasciculated axons that are growing together that mimic the exit of the cells from the retina towards the brain. So now we have the opportunity to make all these different cell types, including that pigmented layer of cells that I mentioned earlier. And the just a movie to show the cells. Okay. So the pigmented cells that I mentioned, the retinal pigment epithelium, it degenerates in a disease called age-related macular degeneration, which is one of the most prevalent neurodegenerative diseases. There's several million people suffering in the U.S. alone. And in this disease, because that very important retinal pigment epithelial layer degenerates in the center, which is the macula, which is where we have high acuity vision. It's the vision that we require for reading, for recognizing faces. Imagine that everywhere you look, there was a big black blob in the center of your vision. How frustrating and debilitating is that? So these patients then have no other disease-altering therapies available, and several groups now around the world are now involved. Excuse me, I just need to go back. In looking at how to replace that retinal pigment epithelial layer. So this is really a global effort using several different stem cell types. You'll see across the top of this table embryonic and induced pluripotent stem cells that are making retinal pigment epithelium. There are other adult stem cells that do not make the RPE but may have trophic factors and trophic benefit that are also being used. And we have a project using an adult or tissue-specific retinal pigment epithelial stem cell that produces a different product from the pluripotent cells, and we're hoping to get into the clinic soon. Several of these programs are already in phase 1, phase 2. We're already getting the early safety data, which fortunately turns out to be very encouraging, and some early hints of efficacy. So there are programs that are being run in Japan, in the UK, Korea, China, here in the US, in Israel. So this is a global enterprise. I think it's a really unique opportunity to see what is going to emerge, to compare the outcome of these clinical studies and these slightly different strategies to see which of them is going to be most beneficial for patients, and understanding that you have a great diversity in the potential recipients. So we may need more than one approach to work for the patients. So we're furthest ahead with this RPE transplantation, but there are other efforts to look at diseases like retinitis pigmentosa and to benefit those with cell transplants. And I just want to point out the use of fetal human progenitor cells, work that's been done by JCite and also Reneuron. They're taking those fetal retinal cells, transplanting them into the eye, and using them either to replace cells or to produce trophic factors that are preventing the loss of the photoreceptors which die in this disease. In addition, over the last couple of years, we've seen exciting progress in gene therapy where the gene is known for retinitis pigmentosa. It has been replaced, for example, RPE65. The product Luxturna that's produced by Spark Therapeutics is now approved and already being given to patients and rescuing vision. So really exciting to see the progress. So just to summarize then, the challenge to produce these cells, I think we've risen to in the research lab, but now how do we manufacture these cells at scale? The RPE transplantation is progressing, but we're still waiting to do more patients to see if it's efficacious, and we should know soon. Photoreceptor transplantation has proven more difficult than anticipated because when we transplant the cells, often they actually fuse or transfer contents with existing cells. Now, that might be beneficial, but it's not the sort of integration that we were initially anticipating. The connection of cells to the brain hasn't been done yet in mammals, so that hurdle in itself is enormous. But we're hoping to see progress there. And with the approval of gene therapies like Luxturna, certainly more are coming along that vein, and we see the opportunity then to put the two things together, to have cell and gene therapy products. The retina's really pioneered so much of this, and I think we'll be seeing that over the next few years. So thank you.
Good. Thanks very much, Sally. David, I will cue this up for you. Yes, I've hooked you up with the laser pointer. You were the pilot, Sonny, sorry.
Thank you, Chuck. Good morning. I'm David Lebwohl, the Chief Medical Officer at Semma Therapeutics. I'm going to talk today about Semma's approach to using stem cell-derived islet cells for patients with type 1 diabetes. I think you know a lot about diabetes. I won't go much into the background, but there are more than a million patients with type 1 diabetes in the United States, and it is a very important health problem for the country. And recently, Semma has several major advances in their work, which will bring us into the clinic using stem cells for diabetes. First are the cells themselves, deriving islets from stem cells. The cells, we've been able to produce a mature islet and produce these in quantities sufficient to be able to be used in the clinic. Now, as Chuck mentioned, the challenge with cells, with allogeneic cells, is that they would be rejected by the body if you don't do something to intervene. And there are three ways forward that we're looking at. The first way is using immunosuppressants, has been used for cadaveric islets, so that you can suppress the immune system and use the islets without any protection at all, to use naked islets. The second way forward I'll talk about is that we've had major advances in the ability to immunoprotect these cells in a device. And I'll show you some details of the device that have made it a great advance, especially overcoming the problem often seen that the body will reject these devices, form a foreign body reaction around it, and starve off the cells. And we now think we have overcome this with our device. The third way forward will be genetic engineering. You heard some about that at the plenary session today. We think this is a very promising approach. I won't talk much about that today, but really talk about the first two approaches, because what we've seen with the first two approaches is what will be reported today, actually reported on Saturday, that we've been able to show that this works in large animals, both the naked cell approach and the immunoprotected approach, and that this is driving our way into the clinic with 2 clinical trials in 2020. So first for the cells themselves. This work comes out of the academic work of Dr. Doug Melton. And Doug's work has now been extended by Semma so that we're able to take a pluripotent stem cell and drive its differentiation into a mature beta cell. These mature beta cells in our program are now of high purity. And what you see here is one way to look at that. This is looking at an actual stem cell islet. You see the picture. With a high percentage of the cells producing insulin. And it really looks very much like what a natural islet would look like. Now, you could also look at these in a much more sophisticated way, either looking at gene expression or looking at the function of these islets. And when you look at each of these look very similar to the natural islets. You can take these stem cell islets and put them into models. And in this case, we're looking at a model in mice. The mice have diabetes induced. So I'll try to use the laser now. Oops. Uh-oh.
You went black. Here, I'll get you. There you go. Yeah.
All right, I won't use the
You
others.
can actually, if you just
Just
click,
here.
you had it
That's
over the
easier.
black
Okay.
button. Yeah.
Okay, so what you see is the glucose goes up because of streptozocin, so you induce diabetes in the mice. And then you put an implant into the kidney of the mouse. Now, these are naked cells being put into the mouse, similar to the way that naked cells are put into people. In that case, it's infused into the liver. In this case, into the kidney. And what you see is a rapid correction of the glucose, coming down to normal levels. You could see there below 180. And you can prove that this benefit is due to your stem cell islets by removing those transplants. You see at those various points, it's being removed. I'll try to get it now. Here, here, and here. And when you remove it, you immediately have a return to the diabetes. So this establishes that the stem cell islets are the ones that have controlled the glucose in these mice. And you can see it actually extends for quite a long while, almost to the lifetime of what these mice are, looking as long as 7 months in this case. Next, turning to immunoprotection. So ideally, you would not want to give patients immunosuppression because they do have side effects. So ideally, you would be able to put the cells into people, protecting the cells, in this case, with a device. So we now have a novel encapsulation method that does protect these cells. And I show some details of it here. First of all, it's a device that has a membrane. You can see the membrane surface here. The membrane surface acts as a filter. The holes in the membrane are too small to allow in the large lymphocytes, but nutrients, glucose, insulin pass completely freely through this membrane. So this allows the kind of protection we want while also allowing the cells to remain alive inside a device. The second key feature of this is the large surface to volume that you see of the device. And not only do you have cells, cells being close to the surface here, but by introducing holes in the device, you allow vessels to grow through the device and also get close to cells that are deep into the device. What we've seen with this is that it integrates very well into the animal so far, and it also, with that, minimizes the foreign body reaction. This foreign body reaction is a reaction that's made it very difficult for any of the previous devices to be successful. Show you again a mouse model of using the device. Here you see again the diabetes induced with high levels of glucose after giving streptozocin to the mouse. The device is introduced, and what you see is a prolonged period of normal glucoses at the bottom, so staying below 180. And again, you can prove it's that the device, the cells in the device that is controlling the glucose by removing the device and seeing that the animals return to hyperglycemia. What will be reported Saturday is that this has worked in 2 large animal models. I'll mention again where that will be. But all this work together has driven our plan to perform 2 clinical trials starting in 2020. And these are the 2 trials. The first trial is for patients who have very hard to treat diabetes, and particularly they have impaired awareness of hypoglycemia. So this is a very severe form of diabetes in that the patients can be, they can go out cold while they're driving or in other situations. And this has been the area where the most use of cadaveric islet transplant has occurred. The advantage that we'll have over cadaveric islets is that our stem cell islets will always be of high quality. It's something we can test, and it can't be assured when you take the cells from a cadaver. And also, they'll always be available when the patient needs them. In this case, the naked cells will be introduced into the portal vein, and the patient will be under immunosuppression with a program very similar to what's used with the organ-derived islets. The second program will go forward without immunosuppression. We'll use the device that you saw that will protect the cells from the immune system, so these patients will not have to have immunosuppression. And in this case, you could treat a much broader group of patients with diabetes. Really, it could be for any patient with diabetes, since we do believe this device will be quite safe for it to be used. So I do want to say where the new data will be presented. The key presentation will be a plenary session on Saturday at 10:30 by Felicia Pagliuca. And she'll talk about a lot of the background data, but also what we've seen in non-human primates, where the naked cells have been shown to function very effectively and reduce the need for insulin in non-human primates. This is the first time that stem cell-derived islets have been able to do this in a primate model. And also, the device has been used in a pig model. This is the human device in the pig model. And here again, in a immunocompetent pig, so a pig who has an intact immune system, we've shown that the device integrates, and the cells are also able to function producing insulin in response to glucose and arginine. Also to say there are other presentations here at the meeting. They're listed at the bottom posters that talk about some of our advances on the cells and in the manufacturing. So thank you, and look forward to your questions in a few minutes.
Good. Thanks, David. And last, I'd like to introduce Vivian Tabora, whom I egregiously misintroduced at the beginning, so my mouth got ahead of my brain, which is to say that her work is actually being nicely commercialized by Blue Rock Therapeutics, of which she is a scientific founder. And if I had engaged my brain, that would have come up first, but it's a volunteer gig, what can I say? Vivian.
Thank you very much. Hi, thank you all for coming. I will just share with you where we stand today in terms of proximity to D-Day, meaning the day we will finally be able to put in stem cells in Parkinson's patients. I think you all know that Parkinson's disease is an incredibly common neurodegenerative disease. It is characterized by significant primary motor symptoms that progress over time to become seriously debilitating. Many of us are familiar with people who experience resting tremors, bradykinesia or rigidity, inability to initiate movement, and postural instability. And there are other motor symptoms, freezing, very small handwriting. And as the disease progresses, other symptoms can become more significant, all the way to cognitive change. Our brains have one main center for dopamine neurons, and that is the substantia nigra, which is in the midbrain, part of the brainstem, where we estimate about 400,000 midbrain dopamine neurons exist on each side. Those neurons project to a part of the brain called the basal ganglia, which works like a relay station that sort of modifies movement as orders for movement are issued in the higher cortex down to the spinal cord. Why stem cells for Parkinson's disease? The only disease-modifying strategies that exist today are a essentially single medical strategy, so giving levodopa, dopamine-like substance, which works for over a honeymoon period for what we call a honeymoon period for a few years, and the surgical procedure where you insert electrodes that dwell in the brain that are particularly effective at preventing tremors and some, but not all, of the motor symptoms. Those electrodes have to stay in the brain for the lifetime of the patient and have a series of potential complications, including needing battery changes every few years, etc. What we hope that stem cells can do is obviously first replace the lost function in the damaged tissues, but also perhaps provide an opportunity for retaining brain circuitry that is otherwise lost when the native dopamine neurons are degenerated. So these advantages could translate into a much more refined and improved way of modifying the disease, as the cells could provide synaptic connectivity and feedback circuitry as they integrate into the brain. We think the cells could provide long-term benefit, and we anticipate that a single surgery in the life of the patient would implant new cells that would then stay with the patient throughout their life expectancy. We also hope that such a treatment could, in the long run, be cost-effective since it's a one-time intervention. Now, speaking of time, to sort of illustrate what Chuck told us early in this conference, we have been working on this, we and of course many other scientists, for a long time. When human ES cells came on board, it took a good 9 to 10 years to finally produce the work I'm citing here in 2011, in which we demonstrated that you can actually make dopamine neurons from human ES cells, that if you were to inject them in the mouse or in the rat brains, they would survive. They would also correct features of Parkinson's disease in these models. We've also injected them in monkeys that are rendered Parkinsonian and have been able to demonstrate good survival, good morphology, as you can see on the lower left hand. One in yellow is a human fetal dopamine neuron, and next to it are neurons derived from ES cells. In animals, in addition to wanting to see that the grafts are safe, that they survive, we like to develop some function. Here's an example of one of the typical tests you do in mice or rats. We're going to see now the rat being pulled, and you'll see that it does not use its left paw. That paw is not paralyzed. It has something that looks like a Parkinsonian patient's bradykinesia, so it doesn't move as you're pulling it. Now we're going to see the same animal after a transplant, and you can see it's using both of its paws. There are similar other tests that we like to see and that we use as sort of a readout or proof of function of the cells, in addition, of course, to examining the brains of the animals. Now, once we were confident in our manufacturing process, we moved to a GMP facility, which we were lucky enough to have at our institution. And there we proceeded to manufacture a large lot of about 1,000 human doses of dopamine neurons. This manufacturing occurred in 2016. We were able to successfully cryopreserve these cells so they can be used on demand and be shipped, conceivably, to different parts of the country. We were able to test their stability so far for about 3 years. And our preset release criteria were met. These criteria address purity, specificity, and safety of the manufacturing process. Having obtained the product, we then started interacting with the FDA. We've had our usual series of meetings with them. It felt very much like this was a learning process for our team as well as for the FDA. But it was overall a very, very friendly, interactive, and useful process, we thought. The FDA required a large set of studies to look for tumorigenicity, biodistribution. So if we put the cells in the brain, do they exit the brain? Can you find them elsewhere in the body? As well as toxicology. These tests required more than 400 mice, and these tests are now finished, and we're happy to say that the product did pass these milestones of safety. The next step was to demonstrate efficacy with the very same cells that we are planning to use in humans. So we used one lot of those 1,000 vials I mentioned and conducted an efficacy study that lasted 8 months with about 48 animals. And we also tested the surgical device that we're going to use to inject the cells in the brain to make sure that the device can deliver the dosing we would like it to deliver. And I think we haven't shown these data much in public yet, but these are the very latest data from the clinical grade product that underwent the efficacy study. So I'm showing data for a test called the rotation test, which is similar to what I showed you earlier, except that the animals rotate here when they're Parkinsonian. As you can see, the animals treated with the grafted cells recovered back to normal, starting at about 4 months or so. No significant difference between male and female animals. And the images are sections of rat brains demonstrating on the left side the control animal. The brown is the dopamine nerve terminals. You can see that this animal remains without any dopamine nerve terminals on one side, and that's because we rendered these animals Parkinsonian. And on your right-hand side, you can see that on the right is the normal dopamine innervation of the basal ganglia, and on the left, you can perhaps appreciate sort of a oval-shaped graft with significant projections into the basal ganglia, demonstrating re-innervation of that part of the brain by the human cells. So obviously, these are exciting data for us, and we are gearing up to go ahead with a phase 1 clinical trial for these cells. This is what the trial will look like. It's an open-label study with obviously primary aim of safety and tolerability. And we have built in secondary aims that will help us see that the cells have survived. We will do fluorodopa PET imaging and, of course, follow the patients for an extended period of time to monitor for their dopamine needs and for their function. The study will use 10 patients. The FDA required that the patients would have moderately severe disease. A challenge for those of us dealing with neurodegenerative disease. When you have patients who are really advanced, it is more challenging to help them than it would be someone in earlier stages. But obviously, for a safety study, it is difficult to argue otherwise. We will transplant 2 doses of cells. Dose finding is a big challenge in our field, since humans have a much larger brain than you can easily model in animals. The procedure, we have negotiated with the FDA that we would like to do this in one single operation, so we can transplant both sides of the brain in one setting. We will need a few tracks on each side to fill up the basal ganglia of the humans. And a similar theme to my colleagues, we do believe that we have to immunosuppress. The brain has been called a privileged organ, but when you perform surgery, you break the blood-brain barrier and you sort of invite immune cells in. So we're going to immunosuppress the patients for up to 1 year and then stop and hope that that would be adequate. This remains a sort of an open-ended question in the field. We will do the surgery with MRI targeting for precision, and having an MRI available during surgery means that we can look at the brain and make sure we did not cause any hemorrhage and that the cells were injected in the right place. I illustrated on the left-hand side where the cells will go. And again, to illustrate how slow things can be, this is our timeline. So 2011, we published this manuscript. We were lucky enough to benefit from a state award. So New York State has something similar on a lower scale to what California has with the CERM. So we were one of few teams that received a $15 million award to accelerate stem cell therapy. That money was used up completely and ahead of time to up to manufacture of the product. And as you heard, the product was then licensed to a company called Blue Rock Therapeutics, which will be supporting the phase 1 trial. The phase 1 trial will occur, the surgery will happen at Sloan-Kettering and our neurologist, and further follow-up will be at Weill Cornell. Here's my disclaimer. So myself and Lorenz Tudor are co-founders of this company. And this is sort of all the, well, some of the people who participated in building this project and some of our funding. Thanks for your attention.
Good. Thank you, Vivian. So at this point, we will transition from presentation to questions and answers. And so I would invite members of the audience to speak up with questions and ask that you use a microphone, please, because this is being recorded. And although you're quite audible, I'm sure, from where you sit to us, for posterity's purposes, we'd like to have your questions recorded. So if you'd kindly use the microphone. Any questions?
Good morning. Kathleen Doney, covering for WebMD. We're doing several stories on stem cells. And I know this is one of those questions you get all the time, but what's taking so long? That's like the average person in Starbucks would ask, or my friends on Facebook.
I mean, I'll start, but then this one I would love to pass down the line as well. It's a new kind of medicine. It hasn't existed before. It's not just making molecules. It's really rebuilding the human body from its component parts. And so nobody's ever done anything like this before. And I think everybody in cellular therapeutics really took a lesson from the world of gene therapy, where we saw the enthusiasm to do good in the clinic get ahead of the scientific underpinnings for the field, and there were well-known complications that happened in the world of gene therapy. And I think everybody here swallowed hard when that happened and said, we don't want to do that in our own field. So let's get the basic science right. Let's build a solid foundation so that when we finally make a first-in-human move, we've done the best possible science that we can. And I never thought it would take 20 years. But if I'd known what I was doing when I started, it wouldn't have taken 20 years. But I didn't know what I was doing. And so, Sally, why is it taking so long?
Okay. Is this on?
I think you're live.
Okay. I guess I have a different perspective. I think it's going quite quickly. When you think human pluripotent stem cell, induced pluripotent stem cells were identified in 2007 and in the clinic about 7 years later, it's extraordinarily rapid. Especially when you think in that time frame, you have to discover how to make the cells that you want. In that case, it was retinal pigment epithelial cells. You have to generate a GMP process. You have to do extensive in vivo studies in animals before you're authorized to move into the clinic. The documentation in itself, it's huge. For our adult RPE stem cell, we had to do a 9-month safety study. That's a 9-month study. And then the reporting after that took 3 to 6 months. That's out of our hands. That's at the hands of a CRO. So when you think of the timeline of how long it takes to make these human cells, develop the protocols, put everything together, I think that we're doing really well. And everyone's excited about gene therapy. Of course we are, but this was a decades-long process to get to Luxturna, and the first approved gene therapies, 30 years. And so same for CAR-T. So I think that we have to do a really good job of educating people just how long the timeline is. And what you hope is that you have a rolling process going, of course, so that each year we could have things that are coming to fruition. But just educating about that timeline is really helpful.
Thanks, Sally. David, would you like to comment further?
Yeah, no, I agree. For stem cell islets, really the same considerations as Sally made. I should say, and some people think sometimes it's the FDA that's slowing things down, and actually what we've seen is the FDA has been very supportive, particularly in recent years, to make these things happen.
Yeah,
Vivian, yes,
similar experience. I mean, after the science was pinned down for us, it still took 10 years because there is a know-how in cell culture that just needs time to, for lack of better terms, to gel. There's a science and an art initially to how you grow cells, and then there's a big scientific and logistical sort of jump that you have to make from, making 1 million cells to treat dozens of rats is feasible, but when you want to scale up for humans, I need several million for each human individual. So there were unexpected scientific and logistical issues in scaling up and then manufacturing to very high level, all of which requires money, expertise, and time.
And as you said, Vivian, this starts with sort of an art. It's people who have a feel for what the cells like, what make them happy, and so forth. But when we started to get seriously into the world of cell manufacturing, what we said is we've got to turn your art into science. We've got to turn your science into engineering so that we can produce pharmaceutical-grade cells that are the same every run, or if not, we certainly know why, and that sort of thing. And it takes a little time.
Thank you.
You're welcome. I have a question for the panel. What's it going to cost? And have you built pricing into the models for manufacturing so that we can treat the masses of humanity who are afflicted as opposed to the elite who can pay out of pocket?
Yeah, I mean, as you know, the scientists don't have any say in the cost equation, but it is a big concern for me. For all of us, I think, in the field. And we all know that, I come from also an oncology background, so I'm well aware of enormous costs for CAR T-cell therapy, for example, and other. I think it might be very costly in the beginning, and these companies who are investing in these products will have eventually to, come to terms with that. For our particular product, it's still too early. It's in just safety domain, so I really don't know. But I think as these projects coalesce and start approaching the clinic, we'll have it will become less, I think, of a one-off. And for them to be viable, they have to be accessible to everybody, not to the few.
David, do you want to comment?
It is early to talk about cost. I should say in terms of the cost of goods is quite high, as you can imagine. But that's also something that with scaling and improvements over time will come down. And I think that will help all these fields. Mm-hmm. Yeah.
How about you, Sally, for the eye?
Yeah, so we're using a tissue-specific stem cell, which means a cell that's dedicated to give the cell product that we're interested in. It's doing its normal job. So the manufacturing is actually very simple. It's one cell culture medium, no need to purify the cells, one passage, and we make a master cell bank. So our cost of goods is actually very low. The biggest part of the cost is actually qualifying the master cell bank, which is done by a third-party CRO to measure things like adventitious viruses, sterility, and so forth. And that is far more than the manufacturing cost. And I think that this is an area, an opportunity for us to put pressure so that we can reduce the cost with new technology. And that is going to benefit everybody.
And one of the ways I think about this is, of course, we're all trying to treat chronic diseases, and the burden of paying for chronic diseases integrated over the lifetime of the patient easily can exceed $1 million. And so therapies, perhaps that are in the hundreds of thousands of dollars, are a reasonable alternative if indeed it's a one-and-done cure. Like we're all swinging for the fence with that. Mm-hmm. Yeah. Other questions? So, David, perhaps you'd comment on. I'm curious about your two-tiered strategies, and in the more brittle patients that you were describing initially, where you're going to the people with lack of glucose awareness and that sort of thing. You decided to go with naked cells without immunoencapsulation. Can you speak to the rationale for that and immunosuppressing those patients?
Yeah, so this is modeled a lot on what those patients are already receiving in not so much the United States, because since the trial has been completed, it hasn't been being used, but around the world. And in the United States, several sites are applying for a BLA using cadaveric islets in that setting. So it is mirroring that. Of course, it's been proven as a way to go forward. So I think it will be very helpful in terms of proving that these islets can replace the cadaveric islets, but come forward with something with higher quality and more availability as well.
Yeah. And Vivian, question for you. You're working with a pluripotent stem cell product like our group is. Do you guys worry about cancer? From, you intend to replace cells, but you get instead cellular overgrowth in some manner. You're a neurosurgeon, right? And so you take tumors out for a living, not put them in. And should we be building in safety switches into our cells and things like that, or is it okay to start this way and learn from our initial patients?
Well, I mean, these are all concerns we had, obviously, going into this. But the experience for our team and many others actually has been that if the cells are properly differentiated, we have simply not seen any cancer. So one should clarify that there's a concern about tumors such as teratoma, which is basically what ES cells would make if they are not fully differentiated. And obviously having that in the brain would be a big concern. And that is not a spot that anybody wants to see themselves or their patients in. So that built into the release criteria is actually, we spent a lot of time looking for expression of pluripotent genes. And so that part we have under control. They're simply not there, and we have not seen a single teratoma to date. We've done in the lab hundreds of animals, and then the CRO just did 400. Whether cells can overgrow, so meaning if they are neural precursors and they're doing what they normally do, which is expand, is what is a concern. And again, the robustness of the differentiation protocol is key to avoiding this. And that's why it takes time. So we put these cells in the animals essentially during the lifetime of the animal, and have not seen concern about overgrowth to date. So, to be honest, when we first started this protocol or this work, we did have safety switches in mind, and we were rapidly counseled to tone that down because you would not be able really to get your product to anywhere near clinical application if you're going to have a lot of gene editing or genetic engineering and building safety switches. The time it takes also to make sure that they are safe, these safety switches, how you integrate them in the cells, et cetera, proved to be really essentially not practically feasible. So we think that the risk is a very small risk that has been balanced by as many checks and balances that we and the regulators could think of.
Mm-hmm. David or Sally, do either of you want to comment on safety concerns that you have wrestled with along the way?
I think it's a very similar story that we've used, refined the protocol, the differentiation protocol, to a point where in testing in animals, now testing, as I said, in large animals and 2 monkeys now, we've put a large number of cells that have never seen a teratoma, which is really the key risk that people are concerned about. So it is refining the protocol to make sure the cells are in a good state.
Yeah. And Sally?
Yes, so our cells are not from a pluripotent starting cell, so we don't have to worry about tumorigenicity to the same level. In fact, the stem cell is not tumorigenic in any of the tests that we've performed. But we are worried about the cell changing in ways that would not be beneficial. For example, like other epithelia, the RPE cells can undergo an epithelial to mesenchymal transition when the cells will acquire a migratory phenotype and they will lose their normal polarized monolayer structure, and they can walk away from their normal layer and actually proliferate in the center of the eye, in the vitreous. So that would not be good. So that is one of the things that we have tracked, and we're using markers to detect any EMT that's occurring. And I'm sure that's a thought as well for others here, that EMT is a very common process that we should all be looking out for. Yeah.
in 5 minutes or?
I have one question.
Okay, please.
And then we're done.
Okay.
So I have a question, I guess, that has to do with how people are interacting and receiving the reports and all the science that's coming forward, because there's a lot of opportunities for people to get good information, and then there's a lot of ways that people can get misinformation about the science and about the way things are progressing. So I just wondered if any of you had a comment about, I guess, or any advice, I suppose, that you might pass on to people with regards to how they could approach the kind of information. And because, if you think a lot of the readers are not necessarily scientists and may not, they can kind of see this as a little bit of the Wild West of, there's all this information out there. Where do we start, and how do we get the right information to make decisions?
Do you want to start, Vivian?
Yes, actually, it's a very good question. One of the things we did very early on, before we even had a protocol written out, is to involve patients, patient advocates, and patient foundations. And we found our interactions with them to be really beneficial to our thinking, and also got us close to sort of how they perceive this work, and allowed an opportunity to distinguish, frankly, between some of the type of work you're hearing about today and some of sort of, let's put some fat cells or bone marrow cells somewhere. And that's a process. I think different organizations have taken that to task. Some publications have done that as well. And we have made a website available to explain in detail what we're thinking about, even well before we had a protocol or a company or anything like that in mind. And I think there remains a lot of work to do to help distinguish. I mean, I think some of the hope or hype questions associated with stem cells have to do with the fact that, people hear about various stem cell approaches that are really, frankly, inappropriate, not scientific, and unethical, in my opinion. So it's a very good point. But when we found the interaction, the patients are very smart. Once you put the information in front of them, they often ask excellent questions, and they have actually helped guide us how to present the information to the greater public.
David or Sally, you want to comment?
Yeah, thanks. This is such an important question, and certainly in the retina regeneration community, we felt it when the U.S. Stem Cell Clinic in Florida injected several people bilaterally with liposuctioned fat from those same individuals and caused blindness. This was a horrific outcome. This was an unproven therapy, so-called therapy, an unregulated clinic. They believed that they didn't have to be following the FDA regulation, which I think the FDA has clearly come out and said that is not the case. And they are in the process, hopefully, of being shut down and stopped, because they're doing this now for Parkinson's and all sorts of different therapeutic applications that they're claiming falsely. And we see not just the FDA, but also the FTC coming in and saying these are false claims. The state's attorneys general also recognizing it. So the more that we're doing to educate the public about these nefarious operations and how dangerous they are, the better. And I'm contacted every week from people who are desperate. Their child is going blind and they want to know what they could do. They've heard about this stem cell clinic in the U.S. or other countries, and they're wondering what they could possibly do for their child. And they are prepared to do anything. They're prepared to mortgage their home and move and really disrupt their lives for this unproven and potentially dangerous therapy and so-called therapy. So I think that what we need to do is to get the word out about that, and the press play a huge role in that. There have been really extensive articles in for example, Washington Post and the San Francisco Chronicle, highlighting these. The pressure needs to be put on so that something is done about it. The International Society for Stem Cell Research has a wonderful website for patients to help them understand the difference between that and between the sort of work that I think we all represent here, which is just a few of the studies and efforts that are following the FDA guidelines or whichever guidelines are appropriate in each area of the country. And so that is called A Closer Look at Stem Cells. Search for that, and I direct people there because they have in lay terms the questions that people should be asking to discriminate one from the other.
And if that was great, thank you. And I'll add just a very brief comment that when we get this sorted and we have legitimate therapies to offer, they're not going to come through your Facebook feed. And they're not going to be wraparound ads on the newspaper because nobody will be able to afford that. Only these predatory clinics can afford things like that. So you will probably hear about them through physicians who have been educated at national meetings through more conventional ho-hum kinds of approaches. So talk to your doctors, I would say, and see what your regular physicians have to say about this, and give a listen to what they have to say. Mm. And with that, I think we'll call this to a close. I thank the audience for their participation and my terrific panelists that we had here. Thank you all very much.
Mm-hmm. Thanks.
Thank you.
Mm.